welcome you all in this class of high throughput data generation and analysis. Students, on the last turn we have studied about what is microarray. So that was the introductory lecture on the microarray. What types of different microarray are there, and means the DNA microarrays, okay, the protein microarrays. So we have discussed about how it is made up, what is the process, okay. So that all was the introductory aspects of the microarrays. Today, students, today we are going to understand what is microarray data. The microarray data is generated from microarrays, okay. By scanning of the microarrays, data is generated, okay, by the automation process. So that data we have to analyze. So what types of microarray data we are going to discuss about that, okay. Okay, this is the schematic presentation of microarray technology. We have also discussed about this on the last turn. So this is the scheme, like probe DNA is taken, or from that the mRNA is isolated from the using that DNA. So mRNA is isolated. This mRNA is then used to produce the cDNA. So cDNA of the mRNA, corresponding mRNA is obtained, and that is dye, that is colored with the dyes, red dyes, okay. So here the upper one is the one sample that is colored by red dye, okay. And another one is the reference solution. In the reference we have taken that we have isolated the gene, and from that we have isolated the mRNA from the corresponding gene. Then cDNA is obtained for that mRNA. That cDNA is then used to make the glass slide microarray. So in the figure you can see this is the microarray slide that is made up of glass slide. We already know that microarray slide is made up of glass slides. So these are two samples, means this is the reference cDNA, and this is upper one is the test DNA that we are going to test. So these both cDNAs are colored with the different fluorescent dyes, red and green. Then these two solutions are mixed with each other, okay. So this mixture is then poured on the microarray slide, okay. So on this glass slide, okay, so we have this, okay. So on this slide we can measure the intensity of each spore, because you can see the different spores are seen here that are arranged in rows and columns. So today we are going to study means how we should consider these spores, what is the authenticity of these spores, how we should consider the spores, okay. So these spores are arranged in the rows and column on the glass slides. So we need to scan, the image is taken, the image is scanned, then it is subjected for analysis by the computational analysis. Statistical analysis is performed in computer. So there are the different colors of spores, colored spore, red, yellow and green, black. So these spores are to be analyzed, okay, to make any inference. So are you able to see this? Students, are you able to see this? Okay. This is the schematic representation, okay. I think there is a disconnection. Okay, join again. Okay, there was some disconnection. Okay, again we are here. Okay, this is the whole process. Analysis involves data mining. So what is data mining? Data mining is to mine, means because here we are analyzing the large number of genes. Okay, so first the glass slide is taken. So we hybridize the DNA and then we wash. So the hybridization process occur there when mRNA is hybridized with cDNA. Okay, so we need to measure how much hybridization occur here. So that is observed by the intensity. The intensity of each spot is important. Okay, so image is scanned, then then it is quantized, means how much intensity is there in each spot. So data mining is used, different data mining method. What is data mining? That is the use of computational method to analyze large amount of data. Okay, in that process we process the image data, intensity of each sports. Okay, then intensity of each sports is obtained in the Excel format. Then Excel format, in that we analyze that data, microarray data. So then visualization methods are used to visualize. Okay, so what is the expression of the gene? So what we get in the Excel sheet, that is the intensity ratio, intensity data. If the spot shows more intensity, then it is thought to be considered that genes are more expressed. Gene shows more expression. There may be low intensity, means that gene shows low expression. So here we can see this is the analysis of the data from microarray 1 in the lower panel and microarray 2. So different genes are there, A, B, C. These are the different kind of genes that show their expression in the different conditions, A, B, C's. Okay, so gene expression microarray 2 and gene expression. So I mean scatter plot. This is the one here means actually regression methods are used to analyze the microarray data. So regression methods are suitable because different genes shows the different types of expression. So it is in the form of scatter plot. Data is scattered, data is dispersed and scattered. Okay, so regression methods are useful in this analysis. Microarray variability in two identically treated microarrays. So microarray 1, microarray 2. Ideally all data points fall on ID line. So this is also called zero line, and this method is called linear regression. So linear regression we use to analyze the microarray data. We are going to discuss about that. Before that we need to know some of the measurement, how did or what is the sources of the error. We need to understand what is the type of data. Before doing analysis we should know what is the type of data we are going to analyze. But this is the popular method, popular graph. So here you can see this is the microarrays, and in the left column you can see this is the microarray slides. So in that we have the genes from different condition, condition 1, condition 2, condition 3 and condition 4. So here we have the 4 conditions. The genes are expressed in the 4 conditions. Okay. So here you can see on the glass slide we have the gene in the lower panel. We have the genes from condition 1, and in the just above the condition 1 we have the genes of condition 2, then condition 3 and so 3 and gene 2 condition 3. So we have different conditions and genes are expressed. Some of the genes may have expressed in the different conditions also, like we have gene that are expressed in the aerobic condition as well as in the aerobic condition. So gene may be expressed in the different conditions. Who is there? So gene may be expressed in the different conditions. So both some of the genes can be expressed in the both of the conditions, some of the genes can be expressed only in the one condition. So it depends. So here on the right hand side we can see the expression matrix. So here on the expression matrix we have the different conditions in the column. Conditions are shown in the different column 1, 2, 3 and 4, and genes are shown in the rows. Okay. So in the rows you can see there are the different rows like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 and 13. So here the total number of genes are 13 and total number of conditions are 4. So this is called expression matrix. What we are seeing on the right hand side, that is called expression matrix, and this is the microarray slide on the right hand side. Okay. Now we are going to understand what is the spores. Whatever spores we are seeing here, you can see on the microarray slide. Okay, how many spores we should consider to make the expression matrix. So here we have to make the selection. We cannot consider all the spores for the expression matrix. Only the genes should be selected that are the spores that are healthy spores that can be selected. So here on this you can see microarray data to an expression matrix. Not the lack of the correlation between physical experimental position of the microarray and the mapping of the data in the expression matrix. So here you can see means you are not able to make the correlation this to this. Okay. So we have to see how we are going to process that. So individual square in gene expression matrix are normally
screen is not visible.
Not visible? Screen is not visible?
Yes, ma'am.
How it can be? I have presented the screen now. Why didn't tell me before that? Now the skin is visible. You were able to see that?
Yes, ma'am.
No, you were able to see at that time I was discussing about this. I have already discussed about this slide. Have you seen this before?
No. No, ma'am.
This is the process I was discussing. Okay. The means how we isolate mRNA, then it is going to be hybridized with cDNA, and this is color be different. Okay, red color. Then this is the green color cDNA from the reference. So these two are mixed with each other. These are then poured onto this glass slide. Okay, this is the microarray slide, and then this image of this microarray is scanned. Then it is subjected for analysis. Okay, and this is the whole process that involves the data mining. Okay, hybridization. We need to quantify that by scanning the image. We analyze the data. Then we quantize how many spores and how many number of genes are expressed in this. So then data mining. We use the one data mining method, one computational method for data mining. Okay, then finally we do the visualization. Using the visualization method, we actually see how many genes are expressed and how much there is the expression of the genes. So we are scanning the large number of the spores are the each gene represent one gene. So we are analyzing the large number of genes at the same time. So this is the popular method of linear regression. Okay, because microarray data is scattered, is in scattered form. So this is the popular method. This line shows the zero line. This line is called zero line, and it is drawn on the 45 degree. So we have different genes that are expressed in different conditions. So A, B, C. Okay. So line is drawn to see the deviation from this. Okay, so each sport, how much there is a deviation. The C is deviated from this line and B is deviated from this line. So deviation we need to see. So this is the one popular method, and this is the microarray slides. Okay. So this is the expression matrix. So expression matrix is derived from this. Okay, this is the original microarray slide, but this expression matrix shows, different color intensity shows the strength of the hybridization. You can see the dark and light color and white color on this, light and dark. So what this light and dark color shows? It shows the hybridization. How much there is the hybridization. So this is the microarray slide. So now today in this lecture, in this study, we are going to understand what is the type of data, which sport we should choose for the analysis. This expression matrix doesn't include all the sports. We need to select the sports. We need to discard some of the defective sports from here. So here you can see there are the genes that are expressed in different condition 1, 2, 3. Okay. So this is the original microarray slide on the left hand side. Okay, and this is the expression matrix that is derived from this. So expression matrix is shown in the right side on. Okay, so in the expression matrix you can see there are the different columns 1, 2, 3 and 4. Okay, and in the rows we can see rows 1, 2, 3. So what are these? These are number of genes. How many genes are present? These are shown in. So you can see here one gene number 13 shows different expression in the different condition 1, 2, 3 and 4. So here you can clearly see one gene low expression, and this is the dark color. In the condition 2 the gene is highly expressed, and in the condition number 3 gene shows the good expression but less than the condition 2. So in the 4 gene show the expression but less than the 3. Okay, and gene is weakly expressed in condition 1. So here you can see by the intensity, color of the intensity, so you can see how much there is the hybridization. More hybridization means more color intensity means it shows more hybridization. More hybridization means more expression, because CD is hybridized with the mRNA. So if there is a more mRNA means more expression, more hybridization means more expression. The gene is the same. Gene is identified with the prop DNA. Okay, that means it shows the high expression. Are you getting what I am saying? Okay, so these all genes are colored with the fluorescent dyes, the yellow, green. So this is the microarray slide. You can see also the colored microarray slide. This is the black and white image. I will also show you the colored image. Okay, now I don't have. Okay, are you getting what is the expression matrix? Do you have any question? Are you understanding what is microarray data?
Yes, ma'am.
This is the expression matrix. This is original microarray slide. Okay, now mapping is done. This is the expression matrix. So expression matrix doesn't include all the sports, but here you can see the different types of sports. Each sport contain one gene. So in this, what happened in this sports? mRNA, cDNA is hybridized with each other. So if there is a more hybridization, that means more expression. Okay, and different conditions are taken. So condition you can see in the expression matrix, you can clearly see. This is the map matrix. Condition 1, condition 2, condition 3, condition 4. So for each gene we can see the expression of each gene in different condition. Condition 1, 2, 3 and 4. For gene number 12 you can see there is a different expression. So what color intensity means? Color intensity, more color intensity means more hybridization. Low intensity means low hybridization. So you can see the expression of each gene vary in different condition. Now we will understand what is the source of variability in microarray data. Okay. Source of variability. So we scan the microarray image. So laser beam is laser beam is targeted on the microarray slide. Then image is scanned. So when we target the spores, so there may be defect. Okay, the beam may not target the spores like this. Here you can see this is the ideal spore. The intensity is focused on this, but here in the B you can see. Okay. Okay, this is not able to focus the spores. Okay, so only half of the spot is scanned now. Here, are you able to see this? So A shows the ideal beam and B shows the beam position error. This shows beam position error. And C shows large beam. Okay, this is the large beam. This may show the false hybridization, false intensity. Okay, because beam is too large. Okay, and this is the laser intensity spores. Okay, so laser beam we are focusing on the each spore to scan the microarray spores. Are you getting what I am saying? These are three types of spores. Okay.
Yes, ma'am.
And here you can clearly see. So A is the ideal type, B is the beam position. It shows beam position array because half of the spores is scanned. So in the expression matrix, if we take this sport, this kind of sport in the expression matrix, so it will show the low hybridization. Even the gene B has, this B has the more hybridization, more expression. This is the highly expressed gene. Then also it may show the low hybridization on the expression matrix. If we consider this type of sport, so we should discard this okay. So these are the error, beam position error, large beam. So A is the ideal spore, so that we should take this type of spores into consideration for the analysis. Now, so the one source of variability. The second, what type of error can occur? Source of variability in this staring method of reading a microarray. Okay. So laser intensity be non-linear across the area of microarray that is excited by the sweat of laser light. So photodetector may exhibit the variation. So photodetector that is used, so that may exhibit the variation in the sensitivity. Okay. So different detector can be used. The detector aperture can vary also. Across the detector aperture, photodetector may exhibit the variation. So here you can see this is the laser intensity. This is his spot you can see. Okay. Sorry. So this is the this is the microarray slide. So here you can see. Okay. But detector sensitive detector means photodetector may show the variation in the sensitivity across the detector aperture as well, means how much it is covering. Okay. So here you can see on this slide, this is not able to scan, means for all the spots. Okay. The laser beam is focused. Now only you can see on the circumference here. The half of the spots are analyzed. Are you able to see this? The spots are not completely covered. Spots are not completely covered. If we focus the beam on the other side, so then also it may leave some of the other spots also. Okay. So photodetector may exhibit variation in the sensitivity across the detector aperture. So this is called okay. So it depends on the photodetector. So this is the laser intensity we are able to see on the left hand side, and on the right hand side detector sensitivity shows that. So it may show the variation. Okay, what is the another source of error? Common source of variability associated with microarray preparation. These sources of error affect both the spotting and steering method of microarray reading. Okay. Microarray spots. Means what happen if the spots are defective. So till now we are talking about the what are the error in the procedure. This is the procedural error. Means laser beam cannot be focused on the spores. Means our spores are healthy, but our there is the error in our method or technology. Okay, here also this is the error of the photo detector. Okay, this is not the error of spores, but we can also have the error in the defect, in the spores. These are the microspores. So here we have the healthy spores as well as the non-healthy spores. So you can see here what type of spores are here. Error source. What is the source of error? So what is the A? A shows the relative location. Okay. B shows variable density. What is the variable density? Means B spores have the variable density. Means in the some of the spores we can have the low amount of DNA. In another spore we can have the high amount of DNA. Means density may vary. Okay. If the one spot has more DNA, obviously it will show more hybridization or more expression, more intensity. Okay, if the spot contains the low amount of DNA, it can show the low expression, but that is the source of error. Each spot should contain the equal amount of DNA. Okay, so density may be vary. And see what variable shape. The spores has variable shape, and in this figure you can see the C spores has the the size of the spores or the shape of the spores is not circular. Okay. Contamination, that is another kind of error. There are some spores which have contaminated DNA, that may have the another gene DNA. Okay. Are you able to differentiate among these four types of error? These are the four types of error related to the spores. So in the A you can see this is show the relative location. Means in the upper panel you can see means all the genes are not at the equal distance. Here you can see A is with this near to this, and this shows more distance to these spores. Are you able to see this? Students, are you able to see this?
Yes, ma'am.
Okay,
yes, ma'am.
because this is the relative location. Means ये वाला स्पॉट इसके कुछ ज़्यादा पास है और ये इससे डिस्टेंस पे है. What is B shows the variable density. Means sample that has been taken in the low and high amount, so it may vary. Or C shows the variable shape, and this type of spores we cannot consider for the analysis. And D type of spores shows the contamination. Means it may have the other source of DNA or contamination. Okay. So these source of error affect both the spotting and steering method of microarray reading. Okay, so these were all types of error we have discussed. Okay, sometimes can be the error from the experimental procedures, laser beam, and these are the different types of spores. There may be the contamination, relative location, variable density problem, variable shape problem. Now how we are going to analyze this? There are a statistical method we can use. The distribution in the special case of normal distribution with mean of 0 and a standard deviation of 1. So we will analyze all those genes. Okay, so we see what is the mean of 0 is standard. So this is the normal distribution curve, you can see. Okay, is the bell shaped. This is the normal distribution. If there is a deviation from this, we can detect the error. It's okay. So now we see how different methods can be used. Statistical, how means what are these, to understand just the type of the data. Deviation from the normal distribution, although statistical analysis of continuous random variable assume in normal distribution. Assume करते हैं कि normal variation होना चाहिए, normal distribution, sorry. Many distribution are not normal, skewed हो सकता है. Here are you able to see? So there can be the skewed distribution. So intensity is depicted here. There is the expected distribution, and this is the skewed distribution. Okay. So it should come here, but it is deviating from this. I mean, it shows the skewed distribution. This is not the normal distribution. And why it is happening? It is due to the errors. We have different kinds of errors. Okay. So we can compare the mean intensity and we can see the distribution. Okay. Continuous random variables. So we can see the distribution. I mean, because there are the different spores and different genes are explained in the different spores. Spores are considered for the analysis. Okay. So what happened? We convert analog signal to digital conversion. How it is done? So now here you can see this is the microarray slide. Laser beam is focused on microarray slide. Okay. Then photomultiplier used. Photomultiplier tube is used here. Okay. Then it produces the analogous signal. Here you can see. You are able to see this is the analog signal. Then this signal is converted into analogous to digital signal. Okay. So here we have the digital signal. So in that we have the resolution, high resolution. Okay. So here you can see. So this is the process how the analogous signal is converted into digital signal. So first the laser beam is focused, then because fluorescent dye is used, so each spot shows the intensity, color intensity due to the presence of fluorescent dyes. Then it is detected by photomultiplier tube. Okay. Are you able to understand this? In the today's class, we are just understanding what is the microarray data. Okay. So here you can see microarray fluorescence statistical analysis. Means how the different intensity we are going to analyze intensity of the each spores. So here you can see we can analyze the using measures of central tendencies, mean, median, mode. Okay, on the y-axis we can see there is the fluorescence signal intensity shown here on the y-axis, and on the x-axis we are able to see the mean intensity, mean, median and mode intensity. I mean only one thing is shown here, I think. Okay, but bars means the height of the bar, what it represents, intensity. So we are only considering the intensity here. Sorry, I mean there is no two different variables. Only we are considering the intensity. Okay. So height of this bar represent the mean means intensity, mean, median and mode. Okay. Are you able to understand? This shows mean, median and mode. Okay. Do you have any question here? This is mean intensity, median intensity, mode intensity. Mode means the frequent value, more frequent value. What is median? जो बीच का value होता है ना. If there is a 5 value, the middle one will be the median. Okay, and there can be the even number, so the middle two number can be taken together. that can be added and divided by 2, then we can have the median values. A mean, and you know how mean is calculated. Microarray sport intensity distribution. So here the different sports are shown: normal, misplaced, mishappen, contaminated, variable. So each sports are shown. Means mean, median, mode, mean and median are shown on the graph. So for normal, we will have the distribution like this. We will have the mean and median ratio like this. For misplaced sports, this can be like this. For mishappen sports, it can be like this. For contaminated sports, it can be like this. For variable density, it can be like this. So we can compare with the reference. If we have the, we can keep this value for the reference. We can have the reference of the bad sports also, contaminated, misplaced, mishappen, and variable density. Using those reference sports, we can easily identify which one is the normal, misplaced, mishappen, contaminated, variable. So we can easily discard those sports from the analysis. Okay, because we know the pattern. We have to identify the pattern. Means by the analysis, how we can identify the misplaced, mishappen, and contaminated and variable density sports. Because now we know this is the pattern of this misplaced, this is the pattern of mishappen, this is the pattern of contaminated, this is the pattern of variable density. So we can easily discard, and this is the distribution for normal, mean and median distribution for normal. Okay, so we can easily identify by seeing this figure. Can you identify now? Can you see the difference among these spots? Are you able to identify the normal? Why don't you reply? Okay. There is the network problem. Okay, so these are the different kind of spores we can identify using the mean and median values. So signal intensities we can identify, and we can identify the contaminated and means error spores, the spores which have the error. So this is intra microarray intensity comparison, statistical analysis of the means of relative fluorescence intensity. So this can be used programmatically, identify contaminated sample that can be discarded from the final gene expression analysis. So from the final gene expression analysis, it can be discarded because we want to discard the defective spores. So defective spores we have to identify. So in that way we can reduce the source of variability by removing the defective spores. Sample 1, sample 2, sample 3. So here you can see the sample 3 contain the contaminated sample. So the profile will be like that. So we can identify this is sample 1 and sample 2. So we can think these are this can be considered for the analysis, and sample 3 cannot be considered for the analysis. So mean is shown here. So this is the mean value on the dashed line. So we can identify the contaminated samples by doing this statistical analysis. Okay. Okay, the lecture was disconnected. Okay, but I have the recording. I will upload it on YouTube. Now lecture is over. So we will study on the next turn about the analysis of these sports. So we'll do some more statistical analysis. So lecture is now over.